I'm thrilled this morning to be able to welcome Dr. Carol Tamminga to our Department of Psychiatry and Behavioral Sciences Distinguished Visiting Lecture Series. As I think most of you know, on this Zoom this year's series, launched a 2-year span of visiting lectures, all by the most outstanding women, mental health professionals and scientists working on psychiatric illness and substance use today. And it is difficult to think of a psychiatrist and neuroscientist who more fully epitomizes that description than today's speaker. Dr. Taminga is chair of the Department of Psychiatry and chief of the Translational Neuroscience Division in Schizophrenia at UT Southwest. She did her medical education at Vanderbilt, then her residency at U of Chicago, where she was on the faculty from 1975 to 79. She moved to Maryland Medical School and then joined the faculty of UT Southwest in 2003, became chair in 2010, and she currently holds the Stanton Sharp Distinguished Chair. Her CV is truly remarkable. She's elected to the National Academy of Medicine in 1998. She's won about every significant honor in psychiatry, including the Lieber Prize, the Founders Award from the International Congress on Schizophrenia Research, the Research Award from the APA, the American Psychiatric Association, Program Chair Award from the Society for Neurosciences, just to name a few. She's published a jaw-dropping 479 peer-reviewed papers at last count, edited 3 books, and despite the demands of leading a large, outstanding clinical department of psychiatry, she continues to PI and co-PI numerous NIH grants. She's a member of the Neural Basis of Psychopathology, Addictions, and Sleep Disorders Study section. She's active on committees in the National Academy of Medicine, and she continues to conduct cutting-edge work illuminating the pathophysiology of psychosis. This research, her work in this area, has really played a central role in the maturation of cellular and molecular science, contributing to understanding serious mental illnesses. The work spans the continuum for bench to bedside. She's leveraged human studies, post-mortem studies, pharmacological studies, neuroimaging, and molecular tools. Among multiple seminal discoveries, including the role of partial dopamine antagonism in treating schizophrenia and identifying mechanisms of tardive dyskinesia, she's been a true pioneer in using multimodal research paradigms to illuminate the role of the hippocampus in psychosis. And she's a leader in the effort to tie molecular and cellular mechanisms to clinical biomarkers, a critical step in developing new therapies. So we couldn't be more grateful that you're joining us. We're sad that it's by Zoom. We look forward, hopefully, to having you back for some face-to-face San Francisco hospitality when the world starts spinning on its axis again. But thank you, Dr. Tamminga, for joining us today. We look forward to your talk.
Well, thank you, Matt, for such a wonderful introduction. I tell you, I'm very proud to be a part of your Women in Psychiatry series. And I just did mention to you that I think it's a phenomenal idea. And I wish I'd thought of it, but since I didn't think about it, I'm just delighted to participate in it. I held off as long as I could, hoping that the COVID would relent a little bit and I could actually come and visit you and see that beautiful new building behind you, but I hope to do it sometime in the future. You invited me to talk about my favorite thing, which is discovering neurobiology and psychosis. Discovering neurobiology is really a possibility these days because people have been working to discover neuroscience now for the last 30 years, and we have some understanding of the brain to work on. So I'd like to tell you what we've done. I'll start first with my disclosures, where I listed almost everything that I do, including the organizations and some drug company work that I do, including chairing a data safety board for Merck, an advisory clinical board for Kinexus, and I'm just a consultant and a stockholder for Karuna. So I love to talk about psychiatry and psychosis, and these are 2 of my favorite pictures. The picture on the left from 1750 is a picture of Bethlehem Hospital, which was one of the first hospitals for the seriously mental ill in London. And if you look at this kind of a scene, and then if you think of the name Bethlehem Hospital and think of how a Brit might say that, the word Bedlam came into our lexicon because of describing the behavior at Bethlehem Hospital. That was in 1750. in 1996 was one of the first articles in an American popular journal, in this case in the National Geographic, where schizophrenia was depicted as a disease of the brain alongside Alzheimer's disease and Parkinson's disease. And you can see there's a great age span, but really the condition of the people with serious mental illness, psychosis, schizophrenia, seems almost no different. Next slide, please. What I'll talk about today, especially starting out, is psychosis. Schizophrenia is more complex than just psychosis, but I'll stay focused down on psychosis, hallucinations, delusions, thought disorder. Next slide, please. First, I'll tell you a story about the bipolar and schizophrenia network for intermediate phenotypes, as we call it, B-SNIP. This is a consortium of people, of 5 sites with 5 PIs, Brett Clements, Elliot Gershon, Godfrey Perlson, Macharya Keshavan, and myself. We've been working together for about 10 years, and we've had 2 waves of funding, what we call B-SNIP 1 and B-SNIP 2. B-SNIP 1, we recruited psychosis probands across the diagnoses of psychosis. And we included the psychosis relatives and about 500 healthy controls. For B-SNIP 2, we focused down on psychosis probands, again, recruiting across the dimension. And we did a deep phenotyping, and I'll show you exactly what we did in just a minute. We had in mind to develop biomarkers for treatment, for biological description and categorizing. What I'll say in the future is that what we were really setting out to do was finding biomarkers for conventional diagnoses. Next slide, please. And you can just put this all the way through, right? When we started B-SNIP, this was the conceptualization that we had. We recruited people with schizophrenia, as you can see on the top line, diagnosis, schizophrenia, schizoaffective disorder, and bipolar disorder. We understood that these conventional diagnoses had fuzzy borders between them. And we also understood that there were symptom dimensions that span these diagnoses: psychosis, disorganized speech and behavior, cognitive dysfunction, negative symptoms, mood symptoms. And these are all just what we imagined. The intermediate phenotypes is what we thought B-SNIP would contribute, these little green round things, the intermediate phenotypes. And then people had the idea that there were genes that would be connected to these intermediate phenotypes, and it would be easier for us to find genes if we did it by phenotype, intermediate phenotype. And then it would be easier to diagnose our conventional diagnoses if we could just send people off for tests, and they would come back, and we could use these to make diagnoses. The reason I show this now is that even though this is where we started, what we actually found is nothing like this. We did not find intermediate phenotypes that fell onto conventional diagnoses with enough power to make diagnoses, and we did not find genes that were easier or stronger connected to intermediate phenotypes than diagnoses. Next slide, please. Let me tell you a little bit about the story about this. So let me go back to the beginning and tell you really what B-SNIP did. B-SNIP collected biomarker measures. What's a biomarker? Overall, these are phenotypic measures, and they're phenotypic measures of patients. Usually we're used to diagnosing by phenomenology. This is an idea that we had to collect cognitive performance, eye movement characteristics, EEG, and then we collected imaging biomeasures too. They were structural MRI, resting state, and DTI characteristics. And what did we do with those? The next slide, please. This is just specifically what we collected to make, what kind of clinical characteristics we collected, along with the SCID and the rating scale, psychosocial function, and then neurobiologic characteristics, the biomarkers. For cognition, they're listed here. Neurophysiology, we collected both evoked potentials and steady state. For imaging, we collected the what I already mentioned, eye tracking, and then for blood, DNA, serum, and plasma, and we have some stem cells as well. Next slide, please. we have a lot of data, as you can see. We have, these are all data from B-SNP1, which shows you the array of data that we have, and almost all of this is published. Next slide, please. I'm not going to go over the specifics of all of the data here, but I'm going to really show you overall what we found in the first B-SNP study. So we were seeking to find biomarkers for conventional diagnoses. So for instance, a person would walk in your office and you would talk to them for 5 or 10 minutes. Then you would make appointments for them, send them away for a week, and have them get the following 10 biomarkers. And they would come back at the end of the week and you would give them a diagnosis. But we were unsuccessful. We did not find biomarkers that fell on conventional diagnoses. And then we had to decide, excuse me, what to do. And it was especially important because we needed the next grant in order to continue the work. So then we thought, why don't we strip the conventional diagnoses from each of the cases, and why don't we recluster by biomarker? And that's what we did, so that we had already identified the multiple variables that differentiated psychosis from healthy people, and that's what we called the biomarkers. And we wanted to understand whether there was a diagnostic uniqueness. The answer to that was no. So then we carried out first multivariate integration in order to develop what we would call biofactors. Then these are biomarkers that run together, but that are unique, complex measures. And then we did a canonical discriminant analysis to ask the question whether with all of these biofactors, all of these biomarkers, we could define new groups with probands and healthy subjects. Next slide, please. This will show you it. This is just really a cartoon of what we did. We looked at schizophrenia, schizoaffective disorder, psychotic, bipolar, and there was a high heterogeneity within these conventional diagnoses that were the same across all the diagnoses. So we, from the point of view of biomarkers, now these people, these individual conventional diagnoses were clinically different from each other, just like we're used to seeing, but the biomarkers themselves were heterogeneous across and non-discriminatory across diagnoses. We pooled them all together, and then we looked at cognition and sensory motor reactivity in order to divide these into what we call biotype 1, biotype 2, and biotype 3. And this division was based on the data from B-SNP1, and I'll show you the replication data from B-SNP2. So biotype 1 is the blue group way on the left, and this is the group that had very low cognition. The cognition levels between biotype 1 and biotype 2 were both very low. The neocortical volume reduction in biotype 1 was the greatest, and biotype 2 was somewhere in the middle and was almost normal in biotype 3. If you look at biotype 3, you'll see a window there where right next to the green band, where you see almost normal resting state EEG and almost normal evoked potential. And using this as a kind of a healthy comparator, you can look at biotype 1 in that little window and find very, very low intrinsic activity, very, very low resting state activity, and low evoked potential. In biotype 2, instead, there's very high intrinsic activity and high evoked potential. Next slide, please. So these are all different from each other. And if you look at how the conventional diagnoses fell within the biotype, on the left in the dot diagram, there's 3 vertical bars: biotype 1, biotype 2, and biotype 3. And the orange dots are the people with schizophrenia. The purple dots are the people with bipolar disorder, and the schizoaffective people are in the middle. And really, the people with schizophrenia, people with schizoaffective disorder are fairly equally distributed, reasonably distributed across all biotypes. The people with bipolar disorder have a higher concentration in biotype 3 and a lower concentration in biotype 1, but still in biotype 1, still 30%. Next slide, please. So these biotype constructions now are multi-dimensional diagnostic. They have certain characteristics, biotype 1, biotype 2, and biotype 3, that I looked at. Biotype 3 started out really to be a little bit of a puzzle because they were quite normal. They had nearly normal cognition, although a little bit low. They were slightly low on EEG power, but still in the neighborhood of normal. Then one of the guys said, well, maybe this is a cannabis group, and we looked to see whether there were differences in cannabis use between biotype 1, 2, and 3, and clearly there was high cannabis use in biotype 3, moderate use in 2, and very low cannabis use in biotype 1. Now, the biotype 1 people might have been so ill that they just didn't go out and get the cannabis. That would also be a possibility. But it's just one of the characteristics of the biotypes that we've kept in mind. So once we had these 3 different biotypes laid out in front of us, we knew there were various tasks. We knew we had to replicate the findings, and I'm going to show you those replication data in a minute. We knew that we needed to do that these had to be good for something, and the thing that we were most interested in is there would be treatment implications to these kind of biotypes, and I'll show you what we're thinking about and what kinds of tests we've started now, and then the genetic characterization, which is for sure. Next slide, please. This is the first of our B-SNP1, B-SNP2 replication data. Down on the left-hand side of the slide, you'll see the items that we replicated, mostly cognition items, and then eye tracking items. The dots in gray are the psychosis people. The dots in purple are the healthy controls. And it's really mostly in cognition that all of the people with psychosis are lower than the normal controls. But you can see the darker of the diamonds, and that's B-SNP1, and the lighter of the diamonds, and that's B-SNP2, both in the healthy controls and in the schizophrenia, and you can really see a very high correlation. We were really surprised to see these kind of correlations, about 0.98. Next slide, please. Then in the EEG, in various paradigms like the paired stimuli ERP standards, frontal ERP, parietal ERP, you see the data in healthy controls in purple, you see the data in the psychosis biotypes in gray, and the solid line is B-SNP1 and the dotted line is B-SNP2. And again, you see very, very high correlation, very high replication in B-SNP2 from B-SNP1. Next slide, please. There's one more picture, which is this is a kind of the summary replication picture. Across the bottom of the slide are the 7 different areas of biofactors. So these are all complex measures. They say BAX, anti-saccade, N100, P300. These are complex measures having not only N100, but the other electrophysiologic parameters whether that correlated with N100 and P300. Again, B-SNP1 in the biotypes is the solid line. B-SNP2 is the dotted line, and you can note rather remarkable correlation. These were 2 studies that were done with not only with an entirely new group of people, but they were done 5 years later. So first we did B-SNP1, and then we did B-SNP2. We function, the 5 different sites that collected these data, we function we try to function as much as possible as a single study. So we maintain inner site reliability, both for our biomarkers and for our clinical interviews and ratings. So really the replication between B-SNP1 and B-SNP2 was higher than we had really imagined. So now the next task for us, next slide, please. The next task was, what do we do with the biotypes? When we went back to NIH, we proposed another kind of characterization a grant, and the NIH said, no, we're not interested in it at all. We want to know if these biotypes are good for anything. So the first thing that we decided to do was a treatment application. And the first, as I'll explain to you why we picked biotype 1, and I'll explain to you why we picked clozapine. But the first test that we decided to do was to make the hypothesis and demonstrate it that biotype 1 predicts the people responsive to clozapine. And I'll tell you why in the next slide, why we developed that hypothesis. Biotype 1, and here you see, that first, reading from left to right, the healthy, you'll see the EEG intrinsic EEG activity picture in healthy controls, biotype 1, biotype 2, and biotype 3. Intrinsic EEG activity is the resting state EEG activity that we measure from a task situation. So it's a resting state EEG, but it's the part of the resting state EEG that's included in the course of a task. So it's an alerted state. People are ready to do a task. And what you can see amply demonstrated here is the very, very low intrinsic EEG activity in B-SNP1 and the high intrinsic activity in B-SNP2. And if you just remember these two pictures, we'll go to the next slide and show you that in this particular data set, we also demonstrated that clozapine, uniquely amongst all of the antipsychotics, increases this factor, increases intrinsic EEG activity. Therefore, it normalizes intrinsic EEG in biotype 1. In that middle figure, you can see the pink graphs. The first, the pink graphs are B-SNP1 and the blue graphs are B-SNP2. You see in the B-SNP1 that are off clozapine, you see the usual kind of reduction in intrinsic EEG activity that I just showed you. And then you see the next very small B-SNP, a biotype 1 effect size, and this is almost normal. And these are the people who are on clozapine. You might wonder, what does clozapine do to people who already have elevated intrinsic activity? And you see that here in B-SNP2 in the blue columns. B-SNP2 shows an increase in its usual high level of intrinsic EEG activity. And then these people on clozapine, of course, just add the intrinsic activity of clozapine on top of that. Risperidone, which is our comparator drug in this study, shows no effect on intrinsic activity either in biotype 1 or in biotype 2. This action of clozapine, next slide, please. This action of clozapine is unique and is consistent with the literature. Clozapine, as you know, is associated with occasional seizure disorders, and it has been reported to increase EEG activity before. We have what we call an attractor network model around clozapine action. You see I've drawn out there just a healthy attractor network. An attractor network is that group of neurons that fires together to represent a memory or an episodic event. And these networks are created with balanced excitatory and inhibitory balances in order to maintain this network. There isn't just one excitation or one inhibition that develops these networks. We think that biotype 1, or we've hypothesized that biotype 1 on just any antipsychotic looks remarkably different, remarkably lower, and really has lost the attractor network configuration. If we put this biotype 1 on clozapine, we hypothesize that will restore this attractor network to normal. And the reason for that is because of the complex actions of clozapine. So clozapine doesn't only have an antidopaminergic action, it has an action at the serotonin, muscarinic, NMDA, and GABA, and probably more receptors. So we've turned to the dirtiest drug to use this. Next slide, please. So this particular study is launched. It's based on the characteristics and the biomarker characteristics that we saw in B1 and B2. The data are blinded and there's no data for me yet to share, but I hope I'll be able to share data with this soon. Next slide, please. There's another way to use these biomarkers, and this is in a particular study that we're doing with Kinexus Medicine, which is a Swedish company, and they have along with Robbie Schwartz, who has described the tryptophan pathway. They have a drug which is an agonist for one of the enzymes in that pathway. And although I'll just try to show you this very quickly in the next slide. So the problem here and why we want to use biomarkers and why this biomarker set has been useful so far is because if Kinurenic acid is a cause of schizophrenia or of psychosis, it's a rather small cause. It doesn't really, dopamine receptor blockade is really something that helps, so that the dopaminergic lesion in schizophrenia is more downstream. Kinurenic acid would be an early lead. And if you use this particular drug focused on kinurenic acid in all comers in schizophrenia, the effect would be diluted out. So we needed a mechanism to find this little blue figure here to find the people with high kinurenic acid. Next slide. So this is all work of Robbie Schwartz, and he has defined the enzymes and the steps in the tryptophan pathway, both in the normal on the left. The pathway goes down to L-kynurenine, and then it splits into an astrocyte-specific and a microglia-specific pathway. And Robbie Schwartz has really looked at all of these enzymes in the tryptophan pathway. He's found out that in schizophrenia, the enzyme that takes L-kynurenine to 3-hydroxykynurenine, this KMO enzyme, is decreased in schizophrenia. The decrease in the activity in this enzyme increases L-kynurenine, and therefore increases the pathway that goes to kinurenic acid. Therefore, kinurenic acid levels are high in schizophrenia, and he's demonstrated both a reduction in KMO, an increase in kynurenine, and an increase in kinurenic acid in human postmortem brain. Kinurenic acid is a negative regulator for the alpha-7 receptor and for the NMDA receptor. So these are both would be pathological features of high kinurenic acid. Next slide, please. I will go very quickly through. Robbie has looked at human postmortem brain normals in the white bars, and schizophrenia in the blue bars, both in the prefrontal cortex 9 and 10, and the KMO is low in each one of those, and the kinurenic acid is high in each one of those. Next slide, please. There's one there's a genetic feature that has helped us really find a biomarker panel for this, and that is that the decrease in this KMO, and therefore the decrease in the increase in kinurenic acid, is only in those people with schizophrenia who have the CC homozygous genotype. So next slide, please. So we really compared the biomarker profile between the CC genotype in the database of people who had biomarkers, the CC genotype versus the CTTT genotype. And we got this kind of a distribution where there were 6 informative biomarkers, of which 2 were low N100, poor facial recognition, and the opposite in the CTTT group. So the plan now, really, in doing this study, is to use the upper distribution of the red violin plot. The upper distribution of this red violin plot along these discriminant scores of biomarkers will include only that very small top of the CTTT group. The reason we wouldn't just use the genotype is that we think there are more cases of high kinurenic acid of low KMO than just that are due to reasons other than just the genotype. So we wanted to catch them all. Next slide, please. So what we're going to do, we haven't started that project yet. The drug still has to go through early safety studies. But when we start the first studies, we'll screen and we'll compose the starting population so that if this drug is active, this is the population it would be active on. Now, I have to stress, just as I'm leaving the discussion of B-SNP, I have to stress that what I've talked about how to use biomarkers in this kind of a setting are all hypotheses. These are translational biomarker fingerprints, and they remain to be established and validated. You know, we've never used biomarkers before. We've used a phenomenology before, and we know how we can use phenomenology in making clean diagnoses, phenomenologically based diagnoses. And we have to go back to the beginning again if we're going to use biomarker-based diagnoses. And we have to show which biomarkers are really informative, which predict treatment response, which predict outcome, which we can use to predict molecular disease targets. I'm sure that different studies you saw from the two that I just showed you, different studies will require different approaches. So stay tuned. I'm sorry, all I have to say now. I wish these studies went faster. I will now talk a little bit about the hippocampus in the next slide. The hippocampus is one of my favorite regions in the human brain. This is a human hippocampus that goes from the anterior side on the left to the posterior side on the right. The human hippocampus is laid out flat so that there's an anterior part and a posterior part. In small animal models, the hippocampus is folded up on itself so that the anterior hippocampus is ventral and the posterior hippocampus is dorsal. We've looked at the hippocampus and I would like to talk a little bit about its association with memory, and then even more than that, its association with psychosis in the next slide, please. This is just to show you that memory actually happens throughout the whole brain, but the hippocampus does something special for memory. In the neocortical areas, there are snapshots of memories taken, whether they're visual, whether they're auditory, whether they're sensory. They're sensory, and then when those sensations, when those memory snippets start coming together, they come into the parahippocampal region, and the parahippocampal region puts them together into context memories and item memories. It's really the hippocampus that takes all of these memories and puts them together into what we would call an episodic memory, an item and context memory, that makes a memory out of diverse kinds of stimuli that come in from various neocortical areas. So the hippocampus really is the region in the brain that pulls together sensory input into a memory and then sends that memory out to the neocortex. Next slide, please. What's very important about the hippocampus is that it's a very well-studied organ. Back in 1950, HM was, we knew that hippocampal hyperactivity was associated with seizure disorders, and intractable seizures were sometimes cured where neurosurgeons went in and took out the seizure locus. HM was somebody who had a bilateral, as you all know, seizure focus. They went in and took out his hippocampus bilaterally, and he lost all capacity to make new memories. He remembered his old memories, but he couldn't make new memories. Since then, since the mid-1950s, people have been studying the hippocampus, so that there's an awfully lot of neuroscience and neurobiology that's known about the hippocampus, and it makes it easier to walk in and say, what does the hippocampus have to do with psychosis? There's a one-way pathway that goes from the entorhinal cortex to the dentate gyrus, CA3, CA1, and then subiculum. These are all stations in the memory process, and each one of these stations does a different performs a different function on memories that are flowing through the process. Next slide, please. The hippocampus, there are hippocampal alterations in psychosis, and these are well known, and I've listed them here. And I just wanted to call attention to the fact that the hippocampus is it works in schizophrenia, but there are indications, some of them subtle, that the hippocampus is abnormal in schizophrenia. Number 3 is the one that seems to be both very, a lot of people have reported it, and maybe a signal of maybe a biomarker actually for conversion to psychosis, and maybe a biomarker for the development of psychotic ideas, not necessarily for other aspects, but for the development of psychotic diseases. Next slide, please. I'll show you what that hippocampal hyperactivity is. And what I'll talk about now is what could be the molecular basis for the increased hippocampal blood flow in schizophrenia. This is the way that a hippocampus looks on MRI. It's the hippocampi sit on the medial aspect of the temporal lobe. In the posterior regions of the hippocampus by the black arrows, the hippocampal there is no hippocampal hyperactivity. The hyperactivity comes from the anterior part. From the anterior part in the humans, from the ventral part in the mouse and rats. And you can see both in the regional cerebral blood flow in a PET study on the left and in an MR study on the right, that there's hyperactivity in the human hippocampus. And not only, these are data from my studies, but from several other groups as well are looking at hippocampal hyperactivity. Next slide, please. What I decided to do in trying to find the molecular basis of this hippocampal hyperactivity is to turn towards human postmortem tissue. And here is again, laid out on its side in a little different configuration, the trisynaptic pathway from entorhinal cortex to dentate gyrus, CA3, CA1, and on out. And what I did that was slightly different than what people had done previously is that I decided that I would look individually at the subfields within hippocampus. I would look individually at the dentate gyrus, individually at CA3, and individually at CA1. What we had really done before this time was merely taken the whole hippocampus, frozen it down, tried to dissect it so that we would have a total sample, a homogeneous sample across the hippocampus. What that did was provide a single sample for hippocampus, but it destroyed all the pathology, as you'll see. So I'm going through the hippocampus now, the subfields, looking for molecular changes, and I'm looking for excitatory and inhibitory molecular changes. The hippocampus itself is a very excitatory organ. It's really run on excitation and the modulation of that excitation by inhibition. So if you're looking for something wrong in the hippocampus, a good place to start is the excitatory receptors. So the first place I went was the dentate gyrus, and the first biomarker that I measured was gluin-1. Gluin-1 is the compulsory. It's the necessary marker for the NMDA receptor. And I saw a reduction in schizophrenia, a very important and significant reduction in schizophrenia in gluin-1. Well, I'm looking for markers of hyperactivity. So a reduction in gluin-1 was really the opposite of what I was looking for. Nonetheless, I went ahead and looked in CA3. And in CA3 was really the gold mine. So we see a reduction in excitatory stimulation in dentate gyrus. And then in CA3, we see increases in BDNF. We see increases in the excitatory NMDA receptor, the gluin-2B receptor, and we see increases in the PSD95, which is the protein that tethers all of the excitatory receptors at the postsynaptic receptor. So these are all measures of excitation. Then Lynn Seliman was, she and I were sitting next to each other at a meeting, and I said, I have this interesting finding in schizophrenia. Let me tell you about it. Wouldn't you like to do some Golgi? And she did Golgi. And Golgi, next slide, please. In this case, Golgi really showed the defining features of CA3 and schizophrenia. In the A frame, what you see are pyramidal neurons. These are human pyramidal neurons. I think they're so beautiful. The apical dendrites go up, and the axons go down from the triangular pyramidal cell body. What you see in B are the TEs. You see the sites of synapses in schizophrenia. Then if you look at C and D, C is a normal apical dendrite, D is a schizophrenia apical dendrite, and you hardly need to do the counts, although we did, and showed a tremendous increase in these synapses. When we looked in E and F at the axonal dendrites, we did not see any difference at all in any of these synaptic constructs on the outgoing dendrites. We just saw changes on the apical dendrite, and on the apical dendrite was exactly where the fibers from dentate gyrus synapse. So that really showed us that at the dentate gyrus CA3 pyramidal neuron on synapse, there was something going on. Next slide, please. And our hypothesis that there was a lesion in dentate gyrus. That lesion decreased the afferent excitation to CA3, and increased the LTP. It increased regional cerebral blood flow in CA3, and that just that increase in hyperactivity was conveyed downstream to CA1 and to subiculum. As it turns out, in our studies, in our animal studies, it doesn't make very much a difference what the lesion is, just that it's a glutamatergic lesion. Next slide, please. We then had to turn to the mouse really to find out anything more from this system. Next slide, please. Oh, well, I'll forget about this. Let's go right to the mouse. Next slide, please. Yes, next slide, please. So we use the mouse, and we now have two models for, next slide, please. We have two models for a reduction in dentate gyrus that causes hyperactivity in CA3. One of these is a dentate gyrus specific gluin-1 knockout mouse, and this is one of Kanagawa's mice that he had characterized partially before, but we took and characterized looking for hippocampal CA3 hyperactivity. And what you can see in frame A is when we looked at the NMDA AMPA ratio, we found really not much difference between the wild type mouse and the knockout mouse. But when we looked individually, like in C and in D, at the AMPA receptor EPSCs and the NMDA receptor EPSCs, we found a very substantial increase in the amplitude, in the EPC amplitude for NMD at the NMDA receptor and at the AMPA receptor. Next slide, please. Which would be the basis of the exact synapse, I guess, the basis of the hyperactivity as it was sent ahead. Then we also looked at CFOS activated neurons in the hippocampal subfield, CA3 and CA1. And we saw a large activity increase, in the number of CFOS positive nuclei. And they're kind of noted by the arrows that you see on the left. Interestingly, what we saw both in CA3 and transmitted downstream to CA1 was we saw this increase in the number of CFOS nuclei only in the ventral part, only in what would correspond to the anterior part of the hippocampus. There was really no increase, no hyperactivity in the dorsal part. Next slide, please. We now have both this knockout mouse, this gluin-1 dentate gyrus specific gluin-1 knockout mouse, which is an exact replica of what we saw in the patients. But we've also used inhibitory dreads in the dentate gyrus, and we found the same kinds of. We have lots of data. We found the same kind of data. The way we put these data together in our model is that there's increased hippocampal blood flow, which is consistent with this mechanism. Many people have looked at this dentate gyrus CA3 synapse, and it's in many both in vivo studies in neurons in culture and in other kinds of animal studies. People have really suggested that this is one of the regulatory spots to regulate hippocampal activity, hopefully to regulate it according to hippocampal need to memory demand, which would be the reason for it. And then for some reason the increase in memory demand in a disorder like schizophrenia and in some disorders like epilepsy gets stuck, and the increase doesn't go away. We think that that the increase in hippocampal blood flow is due to the increased synaptic strength in CA3, increased spines and terminal insertions. The way we think about it is that the hyperactivity exceeds the hippocampal capacity, and false memories, untrue memories, delusional memories are created, and these are then sent out into the cortex and cause unreal psychotic symptoms. Next slide, please. I think I just have one more. Yeah, this is the last of my slides, and then I can take some questions. There are two important observations about hippocampal hyperactivity that we're working on now. One is a linked circuit that we've discovered, and the other one is that there's an adolescent risk window, and I'll point out these one by one. If you look at the figure way on the left, I told you about that yellow figure there, what happens inside the hippocampus and how hippocampal hyperactivity can be started inside the hippocampus. When we see hippocampal hyperactivity and then look generally screening for other areas where there may be hyperactivity or hypoactivity, we see the amygdala, we see the medial prefrontal cortex, and we see the nucleus accumbens as areas that are affected by this hippocampal hyperactivity. We haven't studied these projection areas yet with enough firepower to understand what's happening there. But we do know that there are certainly regions of interest. If you look at the middle is a set of figures, what you'll see there is a contrast between what the dentate gyrus looks like and what CA3 looks like at 6 weeks compared to 10 weeks. So a 6-week-old mouse is clearly an adolescent mouse. A 10-week-old mouse is usually considered an adult mouse, a young adult, but it's considered an adult mouse. We noticed the risk window when we had to pay attention to the age of our animals at which we ordered the animals, because all of a sudden we stopped finding hippocampal hyperactivity if our animals were too old. So then we ran an experiment where we looked at the CFOS activity in dentate gyrus, and that's what you see at the top. And in dentate gyrus, both the dread, now the inhibitory dread that's in a dentate gyrus, produces a decreased hypoactivity at 6 weeks and at 10 weeks. Then if you go down right under adolescent and right under adult, if you look at the control versus the dread, in the adolescent, we see an increase in CFOS, which is the story that I've been telling you. If you take an adult mouse, however, and if you look at CA3 with dentate gyrus inhibition, instead of seeing an increase, you see a decrease. So this would really suggest, and we haven't yet looked into what the mechanisms are that allow the adolescent to generate a hyperactive response and the adult an hypoactive, but we're in the process of doing that right now. I think that's my last slide, other than just showing you what my groups are. This is the group of my collaborators who did all the laboratory studies, the hippocampal studies. And the next slide is the group of people who are all the B-SNIP collaborators who you will recognize, but just in case you don't recognize, I put their names on their picture. So with that, I'll just say that I'm at the end of my talk. Thank you very much for listening.
Thank you, Carol. Really a wonderful presentation of your work. So folks, if you have questions, please enter them into the public chat. And we do have some questions, Carol, and I'm going to take them one at a time. So
maybe you'd ask a question of your own too, Dan.
Yeah, I might do that. So let me start. I have a question from Matt State, and he asks, he says that interesting results from recent genetic studies of schizophrenia show large effects of coding mutations in GRIN2A, but so far not GRIN2B, which is a top autism gene. Overall, the expression patterns are different for these subunits, with 2A being generally later in development. Any findings with regard to N2A and hippocampus in postmortem schizophrenia studies?
Well, as you saw from that slide that I slipped over quickly because it was a whole nother story, we have done a sequencing by subfield in human hippocampus. And we're going to start some sequencing studies now in these animal conditions. I can go look that up. I have to admit I don't have it in my mind, but I don't have it in my mind. I can't answer that question,
okay.
But I do have the data in which to look it up, and I will look it up in the data that we have. Thanks a lot for the question.
Thank you.
Thank you.
So another question from Jennifer Cummings. Is it known how anti-dopaminergic drugs would impact dentate gyrus CA3 activity? And particularly, is it known what clozapine does?
People haven't really looked at CA3 by subfield before, so the pharmacology is, I think we're just gathering those data in the right substance. One of the drug types that has a lot of effect in hippocampus is the muscarinic agonist. If you look at, I don't know that people have directly compared clozapine with other drugs in hippocampal firing, but that's one of the things that would obviously be very, very interesting. And we would suspect, especially because of the muscarinic agonist action that clozapine has, that it might be that it might have a unique action in hippocampus. Dopaminergic drugs definitely have an effect. That may have to do with that loop that I showed, that the nucleus accumbens is affected by hippocampus and is connected to the hippocampus also.
Thank you. Carol, I'll ask one question. I was really intrigued by the data that you showed from the schizophrenia postmortem data in the hippocampal subfields, suggesting increased synaptic connections, which is really opposite to what has been reported over the years that we almost take as an established fact in schizophrenia in the cortex, which is one of reduced synaptic connections, reduced neuropil. Lynn Zellman has contributed to that, David Lewis's group. And recently there's been renewed interest in the idea of over-pruning of synapses as one of the mechanisms. And I'm wondering how you're thinking about the hippocampus and how it manages to achieve kind of the opposite pattern in these subfields. Is there something to be reconciled there in terms of mechanisms?
So I see the hippocampus as a very different organ in the brain than the neocortex. I think that the data on reductions in neuropil and things like that in the cortex are have been very well established. The function of the hippocampus is very different than the function of the neocortex. And even within the hippocampus, the hyperactivity does not exist everywhere. It's relatively localized to the intersection of the dentate gyrus and the insertion of the dentate gyrus to CA3. So what I was explaining was a really almost like a sub-microcircuit of the hippocampus. I wouldn't be surprised if one could find those sub-microcircuits in the neocortex as well, except it's much harder to look there. In the hippocampus, things are much simpler. The levels are simpler, the anatomy is simpler, and I think that allows us to see subtle things in the hippocampus that we maybe would be much harder to see in the neocortex. I don't think that these are inconsistent at all, really. I think that it's just a regional thing. I think that the pathophysiology of schizophrenia is going to be much more complex than we think it is.
So I
hesitate to say things generally in schizophrenia brain.
I so hate to stop this conversation right here. We have run out of time. All the more reason, Dr. Tamminga, that we need to get you here. The plan was for this series to be able to host trainees and junior faculty and have deep discussions. Phenomenal talk. Thank you so much. You've whet our appetite, I think, intensely. There are going to be lots of discussions going on behind the scenes here. Well, we'll look forward to when the pandemic starts to abate, hopefully seeing you here in person. And thank you again so much for taking the time to do.
It was an honor, Chris. I really enjoyed being here. Thanks so much.
Thank you so much.
Bye-bye.
Thank you, Carol.
Take care, everybody.
Bye-bye.
Bye.